And we can introduce our last speaker, who is Maya Wilde, who is a PhD student in Queensland Brain Institute in the lab of Ethan Scott. And Maya, please go ahead and share your screen. And she will present her research on the auditory oddball test for larval zebrafish. And yeah, and as a fun fact, when she's not doing research, Maya loves to knit. And I hope we can also see some examples of what she knits. Otherwise, like, go ahead and thank you.
I didn't realize I was going to be the only one with the fun fact, but yeah, there you go. Now you know something about me. So yeah, I'm going to share what I've been working on, which is developing this auditory oddball test to be used in larval zebrafish, which you've already had a bit of an introduction to with Iris's talk. So the motivation for wanting to use larval zebrafish for me for this experiment is that they're optically transparent. So we can do in vivo non-invasive experiments to see their reactions to stimuli. And we can image the entire brain at single cell acuity and get basically reactions of neurons throughout the entire brain to a stimulus strain, which is very exciting. So we mount the fish in a chamber, as you can see here, and then there's a speaker to deliver the sound. We have two lasers to excite a neuronal fluorescence marker, like GCamp, which shows the activity of the neurons, and then an objective above to measure the activity. And then we can end up with a video like you can see on the right here, showing the calcium activity. So then we segment out the regions of interest, which roughly correspond to individual neurons. And then we can look at the activity of those regions of interest over the course of the experiment and get fluorescence traces like this. So then what we can do is register the locations of those regions of interest back to a zebrafish brain atlas so that we can have the anatomical position of where those different response patterns are happening within the brain and within the auditory processing pathway. So in our lab, up until now, we've done reasonably straightforward analyses of auditory processing in zebrafish. So this oddball paradigm is the next step in complexity, moving along that train. So an oddball paradigm is where you have two different types of stimuli. One of them is presented very frequently as a standard or expected stimulus, and then you have one that is presented less frequently, and it's a deviant or oddball stimulus, which in a human experiment, which is typically done with EEG, the oddball stimuli elicit a mismatch negativity response, where there's an increased response to the stimulus that's not expected compared to the one that was expected. And so when these studies are done clinically, you can see differences in the amplitude and the latency of those mismatch negativity responses in populations like people with autism spectrum disorder. And these paradigms can be visual or auditory, but in my case, I'm going to use an auditory stimulus train with pure tones as the stimuli. So this, like I said, is done in humans with EEG. It's also been done a bit in rodent models, but there isn't a zebrafish version of it yet. So what I'm trying to develop and optimize is whether we can see an oddball response in the brain of the zebrafish larvae. So the stimulus strain that I'm using has two different pure tones. They're represented as blue and red here. So they're two different frequencies. And it begins with a block, which has one of them as the standard and the other is the oddball. And then later on in the train, you can see their roles are reversed. And then in the center, there's a control block in which there's 5 different pure tone frequencies, and each of those are presented at the rate that they would be presented if they were an oddball, but that block doesn't actually have any sort of structure or predictability, so there's no violation of prediction in that control block. So then, rather than comparing the response to 2 different tones of different frequencies, I compare the response to the tone of the same frequency when it is an oddball versus when it is a standard tone versus when it's in the control block. And then separately compare the overall responses to what is here blue and red tones. So separating the ROIs into those that respond preferentially to the higher pitched one versus the lower pitched one, you can see the amplitude of the average amplitude of the responses depending on what the status is of that tone in the different block of the stimulus period. And here, especially on the left, you can see in the high-pitched tones, the amplitude of the response is a lot lower when it's a standard tone, and it's the highest when it's an oddball tone. And you can see an intermediate amplitude during the many standards control block, which shows that the amplitude of the response is increased just when the tone is happening less frequently, but not as much as when it is an oddball, when there's the violation of a prediction of what stimulus would be expected. So this is preliminary data at the moment that I collected to determine whether it's worth going forward with the oddball paradigm in larval zebrafish, and I think that this is sufficient evidence to say that it's worth going forward, seeing as we can see the difference in amplitude there between the many standards control and the oddball, and very different from the standard, the amplitude there. So my next steps moving forward are optimizing the parameters of the control of the experimental paradigm. So I'm using an upgraded microscope and also testing the different interstimulus intervals. And moving forward, when I actually get to do some higher level analysis of these data, I'd like to model the prediction errors. So look at changes in the correlation of activity between different regions of interest throughout the auditory processing pathway, and whether those correlations change following an oddball stimulus compared to a standard stimulus. And then also we have some models of autism spectrum disorder in the lab. One of them is the one that Iris talked about earlier. And I'd like to see whether there are differences in the responses in those compared to the wild types. So a previous paper in our lab did this sort of graph theory analysis where they collapsed down all of the ROIs into a series of nodes and looked at the correlation between those nodes. And you can see where blue is higher correlation, the correlation between the nodes increases with the volume of the tone increasing, but it's also higher in an FMR1 mutant compared to a wild type for any given volume. So I would like to do an analysis like this where rather than different volumes, there's a different role of the stimulus as oddball or standard or during the many standards control block. Great, thank you very much for listening, and thanks to everyone in the Scott lab.
Awesome, thank you very much. Great talk. I have a few questions. If you can go back to the results slide, actually. I wasn't clearly understanding what was the difference between the standard condition and the many standard one. So the many standard is the middle one, and the standard is?
The standard is, so for the blue one, for example, that would be in block 1, where the standard is presented a lot of times. The section that I've outlined there as habituation doesn't go into the analysis, but it's just to establish that tone as being a repetitive expected stimulus. And then I average across all of the ones that are not in the habituation block to get the amplitude of that one.
Okay, and low versus high is block 1 versus block 2?
So that's the different stimuli. So the red here is 531 hertz, low frequency, and blue is 1,189 hertz, which is high frequency. The numbers are all horrible, not round numbers, because it's on a logarithmic scale.
Okay, all right, yes.
Yeah.
Many different like comparisons to digest at once. But
yes,
what was the percentage of the oddball conditions, basically, once you presented like, so I can count like 5 reds and about 20.
Yeah, so it's 20, yeah. So it's 80% standard and 20% oddball.
Okay.
And that is another parameter that can be changed in various oddball experiments, and it affects the amplitude of the oddball response, right? So the less expected it is, so if it's only 10%, for example, then you see higher responses. But at the same time, you want to have a decent number of oddballs so that you can average over several different ones. So 20% is a kind of intermediate in between having at least 5 to average over and then it's still being unexpected.
Because it would be also interesting to see whether like changing this percentage would change progressively, like the distance and the difference between the response for the oddball and the response for the standard in the brain activity.
Yeah.
And see when
and that is,
yeah.
So I was just going to say that is the case in EEG studies that people do in humans. Many people have tried the different percentages and found that difference. Yeah, I haven't really played around with it that much, mostly because I don't want to go any lower than 20, because then I will have less than 5 to play with. But
no, in EEG, usually you use about 25%, I think. 1 every 4 is the oddball.
Right.
Standard.
Can I ask a question?
Go ahead.
Yeah, of course.
Yeah. So, what's the percentage? Is there any percentage differences between high frequency preferring RIs and the low frequency preferring RIs? Are there more cells in one case than the other case?
So it depends a little from fish to fish and with the data that you can get from each fish. So you're not always getting every cell nicely segmented out. So it could be in some fish you just happen to segment the high frequency ones better than the low frequency ones. From memory, there was a difference in this data set. This is just 4 fish, and there were slightly more of one than the other. Yeah, at the end of the day, that's another motivation to not compare responses between different stimuli, right? So
yeah, just also out of curiosity, what's the hearing range?
No, yeah.
Zebrafish?
Well, there's actually a very great paper about to come out from our lab about that, showing, so it's on bioarchives if you'd like to find it. Okay. It's showing that previously people thought it would only be up to about 1 kilohertz, which you can see 2 of my stimuli would be above that. But Rebecca, who's a PhD student in the lab, has shown responses between 200 hertz and 2.5 kilohertz. So it's actually a lot broader than was previously thought. And there's a lot of frequency-specific cells within that range. So ones that greatly prefer one frequency over another.
That's interesting.
Yeah.
And can you actually plot the average response for each of these frequencies during your many standards control to see if there is
Yes, I can. So within those groups that I had defined earlier, these are the preferences within the mini standards control bot. So you can see that one of them, the colors are different on the slide, sorry. So the one on the right, it's actually mostly preferring the experimental tone, but you can see on the one on the left, its highest response is actually to the extra control one, which is of an even lower frequency. And its second highest response is to the one that was actually an experimental one, which is 531 in yellow there.
I see.
Yeah, so, but that is a very good point that, you know, in this case, the neurons that I had grouped together as being the high frequency responsive ones really did prefer that specific tone, but the low frequency ones preferred an even lower tone. Yeah.
And how did you choose these frequencies? Like, were there standard frequencies, or is there any specific reason you chose 355 and?
Yeah, so based on that same research that I was talking about from our lab, Rebecca is the PhD student who's doing that. I wanted to choose ones that were around the middle of the hearing range so that there would be a decent response to them, because it drops off at either end, they don't respond as well. So that's why I chose 531 and 1189 hertz, because the hearing range is best between sort of, 400 and maybe 1300 or something like that. And then, evenly spaced and logarithmically, including the control tones.
Let's see. Because I was also thinking, sorry, but I'm just like brainstorming slash.
That's what we're here for.
That for humans at least, like there is also this individual threshold in which you can discriminate between one tone and another. So if you put 2 frequencies that are too close with each other, then some people would not like distinguish between one and another, or depending on aging, depending on a number of factors. So I was wondering, like, if in zebrafish, like you have like a similar issue, and then maybe if you put the frequencies that are too close, you don't see any oddball because simply the 2 frequencies are too close. It would be interesting to see where is that threshold.
Yeah, so another test that I did early on was actually using bands of white noise instead of pure tones, because white noise for zebrafish is a slightly more naturalistic stimulus. It sounds like splashing in water rather than a pure tone, which is very clinical and experimental. So I divided up white noise into different frequency bands. So kind of high-pitched white noise, low-pitched white noise, and they couldn't tell them apart. So there wasn't any oddball response in that case. So that's why I ended up going with the pure tones. And yeah, as you say, tried to space them apart as much as possible within the range of what they can hear well.
Yeah.